Today we're talking about electrolytes, the charged particles that make the body's electrical system run. Every heartbeat, every nerve impulse, every muscle contraction depends on electrolytes moving across cell membranes in a precise, controlled way. When that balance is disrupted, the consequences can be immediate and life-threatening. By the end of this talk, you'll understand what each major electrolyte does, what drives it out of balance, and how to read electrolyte abnormalities as clues to the underlying disease process, not just numbers to correct. Electrolytes are not just numbers on a chemistry panel. They control three fundamental physiological processes. First, they set membrane potential, the electrical charge across cell membranes that allows nerves to fire and muscles to contract. Second, because water follows osmolality, electrolytes, especially sodium, control fluid distribution between compartments. Third, they are intimately tied to acid-base balance. When electrolytes go wrong, you get cardiac arrhythmias, seizures, muscle weakness, altered mentation, real dangerous clinical signs. The key insight for the system's approach. An electrolyte abnormality is almost always a clue to an underlying disease. Correct the disease, and the electrolyte often corrects itself. Sodium is the master regulator of fluid in the body. Where sodium goes, water follows, which means sodium controls plasma osmolality and fluid distribution between compartments. Hypernatremia almost always means the patient is water depleted relative to sodium. Dehydration, diabetes insipidus, or no access to water. The brain cells literally shrink as water is pulled out of them, causing neurological signs. Hyponatremia means there's too much water relative to sodium, or significant sodium loss, as in hypoadrenocorticism, severe GI losses, or third space fluid accumulation. Brain cells swell. Both extremes can cause seizures and altered mentation, but through opposite mechanism. And here's one of the most important rules in all of medicine. If you have a chronically hyponatremic patient, you must correct the sodium slowly. Correcting too fast causes osmotic demyelination syndrome. You destroy myelin in the brain trying to fix the sodium. Slow and steady. Potassium is the cardiac gatekeeper. The resting membrane potential of cardiac and muscle cells depends critically on the ratio of intracellular to extracellular potassium. Small deviations in serum potassium can have dramatic effects on the heart. Hyperkalemia is one of the most dangerous electrolyte emergencies in medicine. As potassium rises, the ECG changes in a predictable progression. Peaked T waves appear, then the QRS complex widens, then it degenerates into a sine wave pattern, and then the heart stops. This is why the obstructed cat we discussed in Talk 3 is an emergency within an emergency. Hypokalemia causes the opposite problem. Cells become hyperexcitable and then lose function. In cats with chronic kidney disease, hypokalemia is common and causes the classic finding of cervical ventroflexion. The cat literally can't lift its head because the neck muscles are too weak. Chloride is the quiet partner in acid-base balance. It moves in concert with sodium, but its relationship with bicarbonate is the key. Chloride and bicarbonate are reciprocal. When one goes up, the other tends to go down. Vomiting causes loss of hydrochloric acid from the stomach. The chloride leaves the body, bicarbonate accumulates, and the patient develops metabolic alkalosis with hypochloremia. It sounds counterintuitive. The animal is throwing up, which seems like it should make them acidotic, but the acid is leaving in the vomit, and what's left is relatively more alkaline. Diarrhea does the opposite. You lose bicarbonate in the feces, chloride relatively increases and the animal becomes acidotic. Recognizing the chloride change is a shortcut to understanding the acid-base story. Calcium is most famous for bone health, but the small fraction of calcium that's free in the blood is biologically essential for muscle contraction, nerve function, and coagulation. There are two key points about interpreting calcium. First, always check the ionized calcium, not just total calcium. Total calcium includes calcium bound to albumin, and if albumin is low, total calcium will be falsely low, while ionized calcium, the active form, may be normal. Second, hypercalcemia in dogs is a red flag for malignancy. The classic mnemonic, bones, stones, groans, and psychic moans, covers the clinical signs: bone demineralization, urinary calcium stones, GI upset, and neurological or behavioral changes. In dogs, lymphoma, apocrine gland carcinoma of the anal sac, and multiple myeloma are the most common malignant causes. In cats, hypercalcemia is most often idiopathic or associated with chronic kidney disease. Sodium to potassium ratio is one of the most elegant diagnostic shortcuts in veterinary medicine. When you see hyponatremia and hyperkalemia occurring together, which drives the ratio below 27, you should immediately think of Addison's disease. In hypoadrenocorticism, the adrenal glands aren't making enough aldosterone to retain sodium and excrete potassium, so sodium falls and potassium rises. A ratio below 20 is highly suspicious. Importantly, this isn't the only cause. Whipworm infestations in dogs can mimic the electrolyte pattern of Addison's beautifully, as can certain GI diseases. But in the right clinical context, a young to middle-aged dog with vague GI signs, collapse, and bradycardia, a low sodium to potassium ratio should go straight to an ACTH stimulation test. This ratio has saved many Addisonian patients from being missed. Electrolytes and acid-base balance are inseparable. You can't fully interpret one without the other. The basics. The body keeps blood pH tightly between 7.35 and 7.45. Metabolic acidosis means the pH is low and bicarbonate is depleted. The body has too much acid or has lost too much bicarbonate. Metabolic alkalosis means pH is high and bicarbonate is elevated, usually because acid has been lost through vomiting. The anion gap is a quick calculation: sodium minus chloride minus bicarbonate, and it tells you whether the acidosis is from added acids like ketones or lactic acid, or from bicarbonate loss like diarrhea. A high anion gap acidosis means something is adding acid faster than the body can buffer it. A normal anion gap acidosis means the bicarbonate is being lost and replaced by chloride. This distinction narrows your differential list dramatically. Here is your electrolyte workflow. Go through sodium first. It controls fluid balance and osmolality. Then potassium, because hyperkalemia is your most time-sensitive emergency. Calculate the sodium to potassium ratio. It's a 1-second calculation that can tell you whether Addison's is in the picture. Then chloride. It tells you the acid-base direction. Calcium deserves its own evaluation, especially if hypercalcemia is present. Calculate the anion gap if there's acidosis. It guides your differential list. And finally, integrate. Electrolytes rarely tell a complete story in isolation. They make sense in the context of the physical exam, the urinalysis, and the rest of the chemistry panel. Electrolytes are not isolated numbers. They're a reflection of the body's fluid, electrical, and acid-base systems. When sodium is off, think about water balance and osmolality. When potassium is high, think cardiac emergency first and work backward to the cause. The sodium-potassium ratio is a fast screen for Addison's. Chloride points you towards the acid-base direction. Calcium requires a malignancy workup when elevated in dogs. And the anion gap is your guide through the metabolic acidosis differential. Use these tools systematically every time, and the electrolyte panel will start to feel like a conversation, not a collection of numbers to memorize. Question 1. Young intact male boxer, classic Addisonian breed, sodium to potassium ratio of 18, bradycardia, weak pulses. What's the diagnosis and what needs to happen in the next 5 minutes? Classic Addisonian crisis. The sodium potassium ratio of 18 is a big red flag, and the bradycardia tells you the hyperkalemia is already affecting the heart. This dog is in shock. The first thing you reach for is a large bore IV catheter and 0.9% normal saline. Isotonic saline is ideal because it provides volume, sodium, and chloride all at once without worsening the potassium. Get an ECG running. Give dexamethasone IV for glucocorticoid support. Dexamethasone is the steroid of choice here because it doesn't cross-react with the cortisol assay, so you can run your ACTH stimulation test immediately after and still get a valid result. Diagnosis first, then transition to long-term mineralocorticoid and glucocorticoid replacement. Addison's is one of the most rewarding diagnoses to make because once it's managed, these dogs can live completely normal, full lives. Question 2. Significant hypercalcemia in a middle-aged labradoodle with weight loss. Phosphorus is normal. What are your top 3 differentials and which should be at the very top of your list? In dogs, malignancy is the most common cause of significant hypercalcemia, and lymphoma is the most common malignancy causing it. The mechanism is fascinating. Many tumors secrete a molecule called PTHrP, parathyroid hormone-related protein, that mimics PTH and drives calcium up. The parathyroid gland itself is actually suppressed by the high calcium, which is what distinguishes this from primary hyperparathyroidism, where the parathyroid gland itself has a tumor secreting excess PTH. Your PTH and PTHrP results will help sort these apart. High PTHrP with low PTH points to malignancy. High PTH points to a parathyroid adenoma. In a Labrador with weight loss and significant hypercalcemia, get thoracic radiographs and an abdominal ultrasound immediately. Don't wait for the PTH results to start looking for a tumor. Question 3. CKD cat, not eating, weak in the hind limbs, and she can't hold her head up. Potassium is 2.8. What's causing the weakness? Why are CKD cats especially prone to this, and how do you treat it? Hypokalemic polymyopathy is one of the most important complications of feline CKD. The potassium drops for several overlapping reasons. The cat isn't eating, so there's no dietary intake. Polyuria from the failed kidneys washes potassium out, and metabolic acidosis shifts potassium into cells, making the serum level even lower. When potassium gets this low, muscles simply stop working properly. The classic sign is cervical ventroflexion. The cat literally cannot hold up its head. This is dramatic and alarming, but usually reversible with potassium supplementation. Oral potassium gluconate is a preferred route if the cat will eat. If supplementing intravenously, you must not go faster than 0.5 milliequivalents per kilogram per hour. Too fast and you risk causing fatal hyperkalemia. This is one of those cases where the treatment can be as dangerous as the disease if you go too quickly.